Now, methods and resources, I'm not going to say much about that because it will vary so much from one research area to another. But I'm just going to present some overall information and points that I look for when I'm writing materials and methods and when I'm reviewing papers by people from on their materials and methods. There should be sufficient detail to allow a reader to repeat your research. Now, that doesn't mean that they're going to the same field that you grew the plants in. But you need, for example, to provide details of the agronomic conditions that you were using for growing plants. And it means that you either have to write the details of what you did. If it's plants, plant spacing, row spacing, animal husbandry conditions, the age, the stock that you were using, etc. Or if it's been published previously, then you refer to a previous publication where the methods are described in detail. What very often happens is that you have a method that is based on somebody else's research, but you may be modifying it for your own use. So you could say, we use the method based on Briggs and Jones 2003, modified as follows. And then you would give the details of the modifications. I find very frequently, I find that from time to time, that I will see data presented for traits in the results that were not described in materials and methods, and I will also find parameters and measurements described in materials and methods that just disappear in the rest of the manuscript. Make sure that you are consistent in describing parameters and materials and methods, only those that you're going to be discussing and describing in the results and discussion. So there has to be consistency between those two. And in my area of research, which is in the plant sciences, I find it's often quite difficult to be unambiguous in describing how I did the research. This very often requires very careful thought to make sure that other people reading about your methods, they can interpret it in only one way, that there's no alternative way of interpreting it. And I do find that when I'm commenting on manuscripts, there are quite a few occasions when I will comment on their materials and methods to say that it's not clearly defined. And I think one of the areas where there is often a lot of ambiguity is when you're talking about the level of replication. Particularly if you're doing analyses, assays of something that you've extracted from an organism. Because you've got biological levels of replication, you've got assay levels of replication, you've got replication of an extract. You can repeat many different types of activity during the chain from starting the experiment to collecting all of your data. And you've got to make sure that it's very clear when you're describing particularly your statistical methods, you've got to make it clear that what your statistics is referring to. I have in the past been sent manuscripts, not from Serbia, but this is when I was doing reviews for journals. I've been sent occasionally manuscripts where they do their statistics on the assay replicates, not on the biological replicates. So make sure that as far as the manuscript is concerned, that if you are talking about replications, you've got to do your analyses on replications of the biological organisms rather than the analyses that you're carrying out. And that needs to be done unambiguously. Right, I've got some slides coming up which will show you some different ways of presenting data. Because this part of your manuscript is, after all, this is the heart of your paper. This is what it's all about. And this is where the reader's going to be looking at your results to see what they show. And there are many ways of showing them. Present results in a logical order, maybe priority order, which are the most important, or the order, if there's a sequence of events, this leads to this, leads to this. If you measured it, present results for it. If you didn't measure it, well, no, let's say if you did measure it but didn't put it in the materials and methods, then you need to make sure it's in the materials and methods. Design tables and graphs to have the most visual impact, and they must make the points clearly, logically, and unambiguously, and I'll be showing you several examples in the next few slides. Say something in the text about all the points made in the tables and the graphs, and leave your interpretations to the discussion section, unless you are combining results with discussion. So, here are some examples on showing data. Think about the main point that you want the reader to get from showing some information. If you're showing a graph, a figure of some sort, try to identify what is the key point that you want this figure to make to the reader. And then design your figure to make that easiest to see. Now on this figure, I've got two examples of showing the same data. On the left-hand side is one version, and on the right-hand side is the second version. In the extended version of my course, I give the participants an exercise to do, which is designing an experiment to look at different types of sugar. And I've used that as an example of different ways of presenting data in these two graphs here. So here's an example. It's from the sugar experiment that I give, and the primary hypothesis is that different types of sugar will dissolve in water at different rates. So the time it takes for the sugar to dissolve in water is going to depend on the type of sugar. And there was a secondary hypothesis that looked at the effect of temperature. Now, if your main hypothesis is that different types of sugar are going to vary, then use the graph on the right-hand side, not on the left-hand side. Because the graph on the left-hand side makes it easier for the reader to compare the different temperatures, hot and cold. So on the left-hand side, we have hot and cold compared for the 5 types of sugar. On the right-hand side, that makes it easier for the reader to make the comparison between the 5 types of sugar. And there's a separate block of histograms for the different temperatures. So that's what I mean when I talk about focusing on the main point that you want the figure to make. Make sure that is the easiest for the reader to see. Now, this is just filled up with figures. These are presenting results from the same data set. These are examples taken from my PhD students this year. So I gave my PhD students the exercise with the sugar experiment, 5 types of sugar, and I gave them a data set, and I asked them to go away and present results. And this is a range of the results that they sent back to me. It just illustrates how much variation there is in presenting information. So you've got, most of them have presented histograms, bar charts. Some of them, they've clustered the sugar samples together. Some of them, they've separated them into 4 separate graphs. Some of them, they've used a line chart where they've joined the points together. There was one enterprising student, I didn't quite follow what he was doing, but he's just got lines and dots. And then we've got one student has presented them vertically so that you have the bars going horizontally. So you see, there are many different ways in which you can present the information. And I'm not saying that any of those I prefer, or am I? Let me just have a look at one. Let me just have a quick look. Ah, yes. One of them has fallen into the trap. Do I illustrate? No, I don't think I do. So yes, I'm going to illustrate one of them. I haven't got a pointer, have I? No. Oh, I can use my mouse. This figure here. Can you see the one in the middle on the left-hand side? This has got sugar type 1, 2, 3, 4, 5. And the student has joined lines between the sugar types. Now, unless there is an obvious reason why those should be connected, there shouldn't be any connection between those different types of sugar. Because you should be able to use a line joining points to interpolate values in between those points. So if I take a point in between the numbers 1 and 2, what does that tell me about the sugar? It doesn't tell me anything, you see. It has no sense. So make sure that when you're putting your figures together, if you are doing a line graph, you can use the one at the top on the right-hand side, because there, the horizontal axis going from left to right, that indicates temperature. And therefore it makes sense for you to be able to interpolate going down any one of those lines, because that should represent the temperature at that point. So just to illustrate the range of results that you can, or ways of showing your results. Now, my PhD students, a lot of them like to show the data in this format. So present your data in graphs in a logical order. A logical order to the reader, because the reader is not that familiar with what you did. The reader is not familiar with how you collected your data. I presented data to my students for them to analyze in the order in which the bars occur going from left to right. But that's not a logical order for a reader, because if you are able to read what it says, we have on the left-hand side, we have refrigerator temperature, then we have room temperature, then we have hot water temperature, and the one on the right-hand side is 50% hot, 50% room, because I thought, oh yes, well let's mix the two together. So that is what I gave them in their data set. And you see, somebody reading that and looking at that figure will think, well, what's the logical reason for having 50% hot and 50% cold on the right-hand side? So you've got to think logically about how the reader is going to be looking at your information. Does it make sense to the reader? So why are 50% hot and 50% cold samples on the right-hand side? The answer is, that's where it was in the Excel spreadsheet. Right? Why are icing sugar data next to the large crystal data? Well, that's where they were in the Excel spreadsheet. But again, there's no logical reason why it ought to be there in that order. So this order may represent real time, but it's not necessarily logical to a reader. So think about the order in which you present the information. Now, this is an illustration of different ways of showing these same data which bring out a particular aspect of those results which may not otherwise be obvious. Now, if you look at the top figure here, we have a series of histograms. Series of histograms for the 4 temperatures going from left to right in increasing temperature. What I have shown at the bottom is again our 4 temperatures, but this time I've used a line graph. And if you use a line graph in the bottom, you can immediately see that for one of them there's an inversion. The data don't seem to fit the pattern for all of the temperatures for all of the sugar samples. Now, if you show it in that form as a graph, it becomes obvious to the eye. You can see, oh, something's changed. Whereas if you have it just in the form of bars, it's more of a challenge to be able to see when the bars are inconsistent in their size. So always think about looking at different ways of comparing the data before you decide on which is the one that you're going to use for the publication. Now, the next 2 slides, this is from a real manuscript that I was asked to have a look at by some colleagues of mine in Italy. They were doing research on drought responses in wheat, and they were comparing 4 resistant wheat varieties with 4 susceptible wheat varieties to try to characterize the difference between the 2 sets of varieties. And they sent me their manuscript, and this was on 1 page. Here we have data for the 4 resistant varieties, A, B, C, and D, and there are 3 parameters that they've looked at: the relative water content, water loss rate, and one of the amino acids, proline. So those are the 3 parameters that they were comparing for resistant. And on the next page, they have prepared figures for the 4 susceptible varieties. Now, the purpose of their manuscript, the purpose of their research, was to compare resistant and susceptible varieties. So, how easy is it for you to compare resistant and susceptible varieties? Are they the same or different? You don't know, do you? And I think you'll find, any of you, have you seen the handout which I sent? If you try printing out the handout, you will find that those 2 figures are on different pages. So those will illustrate the challenges that you will have in trying to make the comparison between the resistant and the susceptible varieties. So there's an easy way to overcome that problem, and that is to put for each of those parameters, each of the traits that they were looking at, put susceptible and resistant varieties in the same graph. And then have 1 graph for relative water content, 1 graph for water loss rate, and 1 graph for proline. And that makes it obvious that you can compare them. So put the most important comparisons that you want the reader to understand, to appreciate, put those next to each other in the same figure. Now, here's an example of a table that came from a manuscript. Right? Yes? It gets better. Continued. They couldn't fit it all onto 1 page. So my comment that I sent back to the authors was, I can't understand anything in that table, please simplify it. So please keep it simple. Do not try to present a complex table like that, because the reader's not going to be able to understand it. And And you're likely that... do you publish tables like this or write? Of course. Oh, he hasn't got that. I'm trying to think of a good answer to that, but I've got the answer for you. Collaborative research with partners around the rest of the world. You see, you collect 10 numbers, and all your other 5 partners, they collect 10 numbers, and you've got 50 numbers, you see. And it's got international impact. So that's what you need to be doing, all right? But please don't present tables that look like this.